Bom dia a todos, bem-vindos ao quarto webinar da série de webinars comemorativos aos 20 anos do programa Biota. Desculpe-nos pelo atraso, tivemos alguns problemas técnicos para a conexão com o YouTube, mas agora tudo deve seguir conforme programado. Então, esta manhã teremos a apresentação do que o programa conseguiu avançar em termos de conhecimento científico, de relações entre ciência e sociedade, ciência e política, nessa área costeira e marinha. Good morning, welcome all for this fourth seminar that is in the series of the 8 that we prepared for the commemoration of 20 years of the Biota FAPESP Program. Today, we are going to focus on marine and coastal areas and for the presentation of how much the program advanced in terms of knowledge and also in the interface science and policy in this area, we will have Professor Mariana Cabral de Oliveira. She's a professor at the Botany Department, Bioscience Institute at the University of São Paulo, was a member of the steering committee of the Biota program, and she will be joined by Professor Alexandre Turra, that is from the Oceanography Institute from the University of São Paulo, is the chair of the UNESCO Chair on Sustainability in the University of São Paulo, and has been working a lot, not only in terms of advancing the knowledge, but also the links and the use of the knowledge to policies and definitions. So, without further delay, I'll pass the floor to Mariana. Mariana, the floor is with you.
Thank you, Jolie. I will share my screen here. Okay, so I hope you all can see my screen.
Yeah.
And I would like to start by thanking Carlos Jolie for the invitation to be part of this fourth webinar. I also want to thank Vera Cirine and other organizers from FAPESP. And I have to thank two PhD students, Michelle and Inara, who helped me to retrieve and organize the data that I'm going to present today. So I will briefly present a timeline for the Biota program. The program started in 1999. And at the beginning, as part of the original projects, we had two projects that dealt with marine biodiversity. So you will see at the bottom of the slides, these numbers are the ID projects, the ID numbers of each project. In 2001, the program launched an online journal, Biota Neotropica. And in 2002, a subgroup was formed within the Biota program, the Bioprospecta, who deals with natural products, including marine organisms. And towards the end of the decade, there was a major effort to synthesize all the data that was being obtained by these projects in order for this data to be available for public policy making. So there were, the data was deposited in databanks and books were published and maps were organized. And this was actually very successful because there are many legislations and resolutions within the state of São Paulo that use this data that was produced by Biota. So by the end of the decade, after 10 years, there was a major scientific meeting, sort of similar to what we are having now, but of course presential. And in that meeting, the scientists discussed what the program had accomplished by the time, what were the gaps, and what would be the future plans for the next 10 years. And one of the gaps that was actually identified was marine research. So for the first decade, we had only a few, less than 10 projects that dealt with marine biodiversity. So the idea to attract more scientists working in the sea to be part of Biota, what the program did was to make a workshop on marine biodiversity and to launch a project call. And that was very successful because after that initial period, many more projects were started to look at marine biodiversity within the Biota. Another important things that I have to mention was the acquisition of two research vessels, one oceanographic ship and one oceanographic boat. And these vessels sort of improved the access for projects that are working further from the coast and in deep waters. So let's go to some numbers. So this graphic shows for each year the number of projects within the Biota program, and the green part of the bar is what are the sort of land-based projects or projects dealing with fresh water, and the blue parts are the marine. So you can see the initial projects, only two dealt with marine organisms, and for the first decade, not many projects in Biota were looking at the sea. And after this project call, then the number of projects dealing with marine biodiversity increased significantly. Of those 58 projects that we identified as having an interface with the marine environment, we can see that about 1/4 is ongoing. And about half of them are sort of short duration projects of about 2 up to 3 years. And others are long-term 5 years projects. So the thematic and young researchers that have a longer duration of 5 years. And this is the map of the state of São Paulo. And these numbers are the projects where the PI is based. So most of the PIs, the project coordinators are based in what we call the large metropolitan area of São Paulo, but we have researchers in universities inland and on the coast that are also doing marine research within Biota. When we look at the production, more than 750 papers were published in these 2 decades, with an average impact factor of about 2, which is reasonable for the biodiversity area. And these papers were retrieved from the virtual library of FAPESP. And in order for the paper to be there in the library, it has to have in the acknowledgements the number of the project, that ID code that I showed you in the beginning. So if the project don't add that number to the funding or to the acknowledgement part, this paper is not able to be it won't show up in the virtual library. So I do ask, please do not forget to add the ID numbers of your project to your publications. In terms of human resources, of capacity building, about 240 scholarships from FAPESP were directly linked to those projects, and those scholarships include undergrads, technical assistants, masters, PhDs, and postdocs. And we know that this number is underestimated because there are other FAPESP scholarships that are not directly linked to the projects. And also most of the projects have scholarships with federal funding from CNPq and CAPES. So we believe that at least 500 students and researchers were trained over these 2 decades to work on marine biodiversity. So it's a quite impressive number of capacity building within Biota. Also, in terms of project collaborations, there were identified 35 different countries that these projects have collaborations with. And most of the projects had at least one international collaboration. So all this data was, as I said, retrieved from the virtual library of FAPESP. In terms of research areas, this research areas, when the PI is submitting the proposal, he will choose an area that he wants to sort of be linked to. And you can see that most of those projects deal with zoology, with oceanography or botany, chemistry. But it's interesting to notice that there are other fields like education and fisheries and microbiology and even biophysics that are also attracting people from different fields, from different areas. And I think this is very stimulating to be part of Biota because you can get in touch with different areas of research, and this can lead to more interesting collaboration. When we look at the keywords, so this word cloud was built using the keywords that the PIs also fill in the FAPESP form when they submit the proposal. And of course, no surprise, most of the most represented words were biodiversity and marine, but also you can see there are other major focal themes like taxonomy, DNA barcoding, mangroves, natural products, and many other research areas. So in summary, the Biota program in the projects with working on marine biodiversity have been working from organisms from micro biomes to algae, to invertebrates, to vertebrates, and even with human populations living on the coast and that use the ocean as a resource. And as I said, many interesting publications were produced, but I would like to just pinpoint that 140 new marine species were described. And of course, looking at the natural products, there were several metabolites and chemical pathways that were also described. And a vast amount of outreach materials were produced, such as films and interviews, teaching tools, books, leaflets, and many other types of materials. Some of that material you can find at the Biota website. And there was quite a bit of interaction between these projects and the society. And I will finish now and transfer the word to Alexandre Turra, who will give you some examples of that interaction with the society. So thank you very much.
Hello, let me share the screen first. Here. Can you see my screen?
Yes.
Yes. Well, thank you very much for the invitation to share some thoughts on the history of the Biota program. Thank you, Jolie. Thank you, Mariana. Thank you, Professor Luis Eugenio, for the structure that is being maintained in FAPESP, and hope it will be increasing in the future. So this issue is not only important, but it's timely because we have for the next years the decade, the ocean decade, for the science, for ocean or for sustainable development. And then it's really, really important that we have a match or approximation between science and society. And I will bring here some milestones that, in my opinion, contribute to the understanding of the differential of the Biota program. And the Biota program started before it really started. Most of you remember this series of books edited by Professor Joly that created the basis of this program. So it was a kind of overview assessment of what we had in terms of marine, in terms of biodiversity, but also marine biodiversity. And then we started to transfer this kind of information to maps, and then making a very preliminary exercise. We clearly saw that the ocean, the sea, was a big gap in the records of biodiversity. So this was a very strong argument that supported the Biota program. I also would like to mention that this is a very important and strategic aspect of the Biota program. I was there in Intervalles State Park some years ago, learning how to use this very fancy device, the GPS, that was somehow new for most of people that were working with biodiversity. We learned what a datum means. We learned how to put waypoints into this weird device for that time. And then we had the possibility to map in a different way the records of biodiversity in different ways, in a way to start to use this kind of information in other approaches, like, as you saw in the presentation of Mariana, the maps that guided public policy. So the Biota was one of the biggest milestones in this process, and was related to that capacity building workshop we had in Intervalles Park, State Park, about 20 years ago. So it's really important to notice that capacity building is a central aspect of the Biota program. Not only this kind of capacity building, but also this one. I'd like to mention Professor Cecilia Amaral, which in my opinion is a person and a researcher that takes a lot of parts of her energy in capacity building, not only interacting with students, but also producing manuals, guidelines, and materials to people to learn how to identify, for example, the enigmatic polychaetes, but also fostering the structure of museums, collections. So I think this is a very important aspect that was also mentioned by most of you that answered the questionnaire. So this should be fostered, because we need people that know the biodiversity, know how to work with it in different aspects, from museums to the omics aspects of the science. I also would like to mention Professor Álvaro Migoto and his fabulous work in documenting, recording, and communicating the biodiversity. As you can see, the Sifonauta is a repository of images, of fantastic images that makes the communication of the biodiversity easily. It's beautiful, it's amazing, the way we can record it and communicate. This is one example, and this is a very important and strategic part of the Biota program, because we need to communicate. And who will admit that this photograph is not fantastic, beautiful? So it's important to foster this type of skills, this type of approach. Another aspect here, I would like to mention my friend Paulo Sumida, professor at the Oceanographic Institute, that is looking for new frontiers. As you can see, he has only one t-shirt because he loves two things in the world, not more than that, okay? But he loves the ocean, the deep sea, and also Palmeiras, which is his team. He is a supporter of Palmeiras. And he won some prizes because he took photographs of wearing the t-shirt of Palmeiras in different environments. But this represents an important effort to discover, to investigate new frontiers like the deep sea, like Antarctica, like other, even closer to us, like the southern coast of São Paulo, which for some areas is a frontier. And then I'd like to mention the work and the passion of Paulo Sumida to the ocean, to the deep sea, to Antarctica, and to oceanographic as a whole. And he is launching this book, Brazilian Deep Sea Biodiversity, soon. It's a series published by Springer, which I am editing with them as a subproduct or a side effect of the Rebentos project, which was also linked to the Biota program. So soon we will have this amazing book being published. We also have new frameworks. We are talking about new ways to look at biodiversity. This scheme here talks about the linkage between biodiversity and ecosystem services and human well-being, or the benefits that nature brings to society, to people. And then we can put the biodiversity into a different context, into a new framework, when we will look obviously at the species, at the communities, but also into the ecosystems, and understand how they are structured, how they function, and what are the benefits to society. In a way, we establish a new form, a new way to communicate with decision makers, with society, and reinforce the importance of biodiversity. So this is really, really important, and it's also it's a milestone that we realize in the history of the Biota program in some of the projects, mainly the recent ones. We also have new dialogues. So the Biota program created the opportunities to link biodiversity to society and to establish dialogues with the users or the managers of the environment. So this is a project that was coordinated by Professor Cecilia Amaral from Unicamp, focused on a small bay in the São Sebastião Channel, where we had that opportunity to put in practice and learn a lot with this interaction with local stakeholders in a way to use biodiversity, to put biodiversity into an applied context. So I will bring some of the results of this project, not numbers necessarily, but the type of product that are useful not only for science, but also for society. This is a book on methods to study this kind of environment and to conduct this kind of work, which is an integrated and interdisciplinary work. So in this book, we try to capture the complexity to work into this new realm of integrated and interdisciplinary research. This on the right hand side, we have the book Life in Araçá Bay. We also have in English, to show, at least for the mayor of São Sebastião, that Araçá was alive, and it is and will be alive for a long time. There are some products, sorry for the definition of the images, but there was a very strong effort in bringing the biodiversity closer to society, like these leaflets, folders, and books that were produced by specialists and that were received in a very excited way by the local users, the local society, even children that took these materials and started to look at Araçabe to identify the birds, to talk about the trees, talk about the fish, and it was really an amazing point that represents the desires, the wishes of some of you in terms of communicating science. There were other products that were more linked to the idea of the socio-ecological systems, of this socio-ecological system that was Araçabe. And then we started to discuss how to integrate the science, the processes, to discuss the future of the area, and then the public policy. So the plan, the local plan for the development, the sustainable development of Araçabe was one of these products that created a baseline to solve the problems of the bay. And then I consider these products have a very important role in bringing the university closer to the society, the science closer to the society. This is a documentary that was produced based on the findings of the project, interviewing local people and trying to capture the importance of the bay, the importance of biodiversity, of local traditional knowledge, and the other importances of the bay. In the end, we try to capture the importance of this kind of approach, this interdisciplinary and integrated research in a paper that was published in Ocean and Coastal Management. As you can see here in the left-hand side, this was the social network of the project in the beginning. The number of lines and the width of lines represent the connections between modules. Each dot here is a module, which talks about different aspects of Araçabe, and so the number of arrows and the width of arrows represent the integration. So when we ended the project, you can see how integration was fostered by all the initiatives we did in the project, like workshops, like meetings, publications together, and that. So we can do that. We are learning how to do that, and Biota program is supporting us to do that. So there are some legacies of the Biota project in terms of lessons learned. How do we foster oceanography? How can we support decision making, and how we can interact a little bit more with society. This was somehow a starting exercise, a starting project, so that we can learn based on it and move on. I will not go into deeper details here in describing the legacies, because we have another talk that will make us think a little bit more on the importance of the Biota project, but also in the future. So let's think what comes next, and this depends on us, on what we want to do, and how we build the Biota program together with FAPESP. So thank you. That was the message.
Thank you, Alexandre and Mariana, for presenting this summary of the achievements of the Biota program in this area of coastal and marine biodiversity. And now we have a pleasure of having Professor Derek Tinteser from the Department of Biology at Dalhousie University in Canada. Derek has been working with macroecology of oceans, marine macroecology. He also published a very good book, the theory of biodiversity, and he was going to present us a talk, Developing New Models to Understand the Process of Global Patterns of Marine Biodiversity. Professor Tintesor, the floor is with you.
Thank you very much, Carlos. Firstly, I want to thank the organizers for giving me this opportunity to talk to you. And secondly, I want to congratulate you on achieving 20 years with your research program. I think that's a great achievement. And in my experience, the research programs that continue the longest are the most valuable ones. So I think it's a great achievement, and I hope you achieve many more exciting findings with your research program into the future. Can you see? I'm hoping you can see my screen and can hear me okay. Please let me know if that's not the case.
Yes.
But what I want to talk to you about today is in some work that I've done on trying to develop models to understand really the processes, the ecological and evolutionary processes that shape the distribution of marine biodiversity around the world today. Before I do that, I want to say I love Brazil. I love your country. I really enjoy all of the working with all of the Brazilian researchers who I've met and collaborated with. And I know you face some challenges with COVID and with the environment, your incredibly biodiverse environment. But I think you have a great country, and it's great to see the research that's going on. So I just want to start by thinking about how ecology has evolved over time. So really, we've had at a large scale descriptive ecology since the 1700s, and of course, Alexander von Humboldt was the first person to really describe the gradient of biodiversity from high biodiversity in the tropics and near the equator to low biodiversity near the poles. But really, analytical ecology started more recently, maybe in the early 1900s. But often we have generally small-scale observations and experiments. Most ecological studies are on the meter scale and last less than 5 years or so. Not all, but most ecological studies are small scale. However, as we get more and more data, we start to confirm some of these earlier observations about gradients in biodiversity. So Kevin Gaston called the latitudinal biodiversity gradient Earth's first order biodiversity pattern. That is the oldest described pattern and the most obvious one on our planet, in the sense of high numbers of species, again, in equatorial regions. But it's been a real mystery as to why this gradient exists. Not just a mystery, but there have been many, many explanations proposed for this gradient. In 1992, Rhodes suggested over 30 explanations, some of which he argued were circular, but the exact mechanism remains elusive. And part of the problem for that is that one potential driver, such as temperature, can operate through multiple mechanisms. So it's very difficult to separate out a statistical relationship between temperature and diversity into a mechanism, or to separate out the potential mechanisms. So you end up with big tables like this. This is not for you to read, but just to show you that you can get these complex tables of these range of hypotheses as to explanations for the latitudinal diversity gradient. And then again, relatively recently, we started to put together these spatial patterns of the distribution of biodiversity across the Earth. And certainly the first one I've seen in the marine environment is actually in a geological journal by Staley et al. In 1967, describing the global diversity patterns in bivalves. And some of the features that we see here, such as the hotspot of biodiversity in the tropical Indo-Pacific region, still remain in the modern day discussions of the spatial patterns of biodiversity. I'm not totally sure when the first spatial descriptions of terrestrial biodiversity were, but I'd be interested if you know more on that subject. And then in the 1990s, people started to take some of these large-scale patterns and analyze them statistically. So distill these large compilations of data, fit statistical models, and try and hypothesize what drives the abundance and distribution of species at large temporal and spatial scales. But as I mentioned, part of the issue is that, and we're all guilty of this, I do this, right? Everyone does this. You fit a statistical model to data, and you derive a pattern, such as here, the relationship between sea surface temperature and normalized oceanic diversity, and you find a relationship, and then you hypothesize what the mechanism might be. But that statistical model does not necessarily allow you to distinguish between these mechanisms, because different hypotheses can each predict the same pattern. And that's just a limitation of correlative models. So is there a way around this? Well, obviously we have one replicate, right? We have one planet Earth. We can't perform replicated experiments at large scales or at global scales. So the only way we can do that is by developing models. And the kinds of models that I'm talking about here are now process-based models. So these are simulations of how biodiversity evolves over time. So you encode the processes, the interactions among species, evolution, death, dispersal into a model, and then you let the species and populations evolve and see how they evolve over time and see what patterns they generate. And if you encode different hypotheses into those models, you can see how different the patterns are that they generate. So then the question is, can we do this at a global scale? Is it possible to do that at a global scale? And can we do it across very different environments? Now, as a marine ecologist, I focus on primarily on marine environments, which are very, very diverse. So you have your high diverse nearshore environments. You have your oceanic and pelagic environments, which operate under very different conditions, very few constraints on dispersal, for example. And then deep sea environments, which are very stable in terms of their environmental characteristics, very food limited. And of course, terrestrial environments, which are also very diverse. So the caveat of the work that I'm going to talk about is that it's very scale dependent. Okay, these models are not intended to derive what drives diversity at small scales within habitats or within small sites. Obviously, different factors are important for structuring biodiversity at different spatial scales and different resolutions. And here I'm looking at large scales and coarse resolutions. So going back to what Rhodes said, there's 30 or more explanations for these gradients. So what can we do? We can look at the empirical data. We can look at spatial patterns from the literature and try and get clues about what drives this biodiversity. And I'm actually going to start with terrestrial environments. So if we look across a number of different taxa, birds and amphibians for example, show relatively similar patterns. Again, hotspots of biodiversity in the tropics. There's a combination of productivity and rainfall that is highly correlated with patterns of biodiversity. Plants, again, a similar pattern. So you can see that there's a very consistent relationship across quite different taxa. If we look at mammals, the top panel shows richness, the bottom panel phylogenetic diversity. Again, the hotspots, the darker colors, are in South America, in the tropical regions, but also generally across the tropical band and then declining towards the poles. So just very briefly summarizing that, we can see that diversity is strongly correlated among these quite different taxonomic groups, with broad tropical peaks in diversity. And most patterns on a large scale are related to temperature and to rainfall. On smaller scales, productivity plays more of a role. So now I'm going to go into the marine groups, which perhaps some of you are less familiar with. But if we start with coastal species, then what we see, and again thinking back to the bivalves described in 1967, is a pattern that's quite similar. So if we look at coastal fishes, coastal sharks, and non-squid cephalopods, we see again a hotspot in the Indo-Pacific region, sometimes a secondary hotspot in the Caribbean or the Caribbean area. Fairly high diversity in the Indian Ocean. Again, red is high diversity, blue is low diversity, and then a decline from the tropics to the poles. And that's the same for most of the coastal groups that we look at: corals, seagrasses, mangroves. But if we look at pinnipeds, so if we look at seals and similar organisms, we see an inverse pattern. So there are groups which are specially adapted to particularly cold environments, but on the whole, the relationship is similar. And then if we look at oceanic species and pelagic groups, we see a slightly different pattern. You can see it most clearly in the tunas and the euphausids, planktonic euphausids. What you see is kind of a broad smear of diversity across the tropics and the subtropics, perhaps with a hotspot in subtropical regions instead of tropical regions, perhaps a bimodal distribution of biodiversity, and the same for cetaceans, for whales, similar. So we see that this environment where dispersal is a lot less constrained, we see a slightly different pattern. And then the deep sea, of course, is a wholly unique environment. And for this group, we looked at ophiuroids, or brittle stars. And it's really interesting because in shallow waters, so at the top, from 0 to 200 meters, you see a pattern that's very similar to all other shallow water groups. But then as you go down to kind of mid-depths, 200 to 2,000 meters, it starts to move away from the tropics. And then when you go into the abyss, into the deep sea, you see an entirely different pattern. So hotspots of diversity along coastal regions and at high latitudes in particular. What's behind this probably is the export productivity, so the rain of food that's coming down to depth. The distribution of ophiuroids correlates quite well with the export productivity, so that there's a real food limitation. So in the marine environment, we see three distinct patterns. We have your typical tropical peak for coastal species and in the equatorial Western Pacific. Pelagics have a much more smeared distribution, and then deep sea species peak at high latitudes. But again, temperature is a really strong predictor of diversity and habitat availability as proxied by coastline length or frontal systems. And then deep sea richness seems to be driven by export productivity. So we can put all this together into a model or a theory. And if we're looking at the distribution globally, then the fundamental mechanisms should be consistent across different environments and across different taxonomic groups. Our temperature is the only factor that connects all patterns except for cold water species. And then you have additional constraints that are different based on which habitat you're in. So terrestrially, it's water availability, it's a limiting factor. In coastal systems, so for coastal species, it's the length and complexity of the coastline. You have frontal systems in pelagic zones and export production in the deep sea. So then how do we model the actual processes that are responsible for, or potentially responsible for, these distributions? Well, there's a nice paper from 2010 in the Quarterly Review of Biology where Mark Velland suggests that there are just four key factors that set diversity at all scales. There's speciation, drift, selection, and dispersal. Dispersal within and between these different scales. So our approach was to start with these fundamental processes. So we start with a neutral community in which all species are just considered identical. And this has drift, dispersal, species dispersed from local communities, and random speciation events. We then add a thermal gradient with using metabolic theory to predict how interaction rates and speciation rates both increase with temperature. We add a habitat gradient. For the marine environment, you get declining habitat area from the tropics to the poles. Obviously, that's not the same in terrestrial environments at a global scale. And then we add niches. So niches bring in the selection based on the characteristics of the organisms. So I don't want to go into a lot of detail, but just in case you're not familiar with neutral theory, Stephen Hubble's very controversial theory, he assumed that all individuals are identical, no matter their species identity. And this applies within a trophic level or guild. And then there's assumption of zero-sum dynamics, which means that organisms are competing for a limited pool of resources. Now, neutral theory, no one believes that the real world operates like that, but it provides a really good null model of ecology. If you can reconstruct patterns just with neutral theory, then why do you need to invoke other factors to support them as drivers of biodiversity? So it's a basic model that you can then compare other factors against. And again, the metabolic theory of ecology from Jim Brown suggests that metabolic costs are proportional to temperature. So it can operate as per the diagram on the right. So the temperature affects metabolic rates. And in evolutionary terms, this can affect mutation rates, potentially speciation rates, potentially extinction rates, and then ultimately species richness. It can also affect ecological processes. So the interactions and the rate that organisms interact are increased at higher temperatures. Both of these mechanisms are encoded in the model that we created. So just to give you an idea of this model and its structure, you can imagine the globe as a grid, divide the globe up into a grid of cells, and then we can unwrap that grid to give you an idea of how the model works. So the model is a set of local communities, and these local communities can be small or large based on the amount of area. And they consist of a set of individuals of different species. So here the colors indicate different species. And they randomly interact with one another. Every time step something dies and is replaced, organisms or populations can speciate. They can disperse either within the local community or outside to the larger communities. And we can also impose a thermal gradient. So hot at the equator and things interact faster and speciate faster, and cooler at the poles. And then the idea is we start with a completely random community random species distributions, and we run the model and see how the patterns of diversity evolve over time. Again, individuals die each time step. They're competing for limited resources and for space. And all of the patterns that we see are not fixed. They emerge over time through populations. They grow, they speciate, they go extinct, they disperse, and so on. And then we start to impose some of the real world conditions that we see. So we start to use realistic climate conditions, have continents superimposed, so you have these boundaries on where species can disperse to. And then we start to include gradually these metabolic, habitat, productivity, and niche components. And then what we do is we encode these patterns into a model which ultimately has very few parameters. It only has 3 or 4 parameters, and see what patterns come out. So you start with a null model, which essentially means all species are equal. They just disperse at the same rate. They speciate at the same rate. And if you start with a random community, as you can imagine, you end up with a random community. So again, richness, red is high richness, blue is low richness. And then we start to add different aspects. So we can add observed continents and distribution of land masses. And as you can see, that changes the distribution and the patterns that emerge. But it still doesn't look anything like the real world. What if we start to include habitat effects on community size? So habitat area. So for coastal organisms, this could be coastline length. For pelagic organisms, it would be frontal systems. And again, in areas with more habitat, you have larger communities. And we start to see more distinctive patterns emerge from the model. Again, this model is starting with a random community, and just the patterns emerge over time. Then if we start to add thermal effects on speciation, so things speciate faster in warmer environments, then we start to see a more realistic gradient of biodiversity evolve. Again, quite smeared out, but reasonably realistic. And then we can combine area and thermal impacts on metabolism and on speciation rates. And finally, we can impose niches. Interestingly, niches don't affect the pattern that much. You can get a reasonable pattern without invoking niches, but they are more important, perhaps counterintuitively, in high dispersal systems like the pelagic, where without niches, things could really disperse in an unlimited capacity. They could disperse all around the oceans. So niches have more of an effect in these high dispersal systems. So we created this model, and then the next thing to do was say, okay, given this, how well does it compare to empirical observations? So we looked at empirical data. So on the left, we have the empirical observations normalized to between 0 and 1. And we plotted the latitudinal gradient. On the right, we have the simulations. This is looking at coastal ectotherms. And we plot the simulated latitudinal diversity gradient, and then we look at the spatial correlation. So you can see that in this example, the model, if you simulate it and if you run the model and it's stochastic, so you might run it 1,000 times and look at the mean results, they're very similar. And in fact, the latitudinal gradient is very similar to the empirical data. If we look at pelagic systems where you have a much more smeared out distribution of biodiversity, again, we see, in fact, a better correlation of the spatial pattern and the latitudinal gradient. In the deep sea, the model doesn't do quite so well. We do include export productivity, regulating community size. The correlation isn't as good, but the gradient that emerges is reasonable. And then finally, we applied the model on land using productivity as a proxy for habitat area, but also using temperature and temperature to regulate speciation rates. And surprisingly, the model even does a reasonable job of reproducing some of the terrestrial patterns that we see. So just to summarize these results, here we have a mechanistic model that simulates global biodiversity from random starting communities. We find that there are two specific mechanisms that predict global patterns in surprising detail. So a metabolic scaling of speciation rates, a metabolic scaling of species interaction rates, so ecological interaction rates, doesn't really affect the pattern. So it can distinguish between those two temperature effects. And then community size scaling with habitat area and productivity. So resource availability helps to match to observed patterns. And we find that niches are very important for local species identities. So for setting the identity of species that are within a system, but over evolutionary timescales, over really long timescales, they are generally not as relevant for large-scale patterns. And really, this is a first-order model looking at these large-scale patterns and processes. So I just have a few more slides. A lot of what I just presented is discussed in this book, A Theory of Global Biodiversity, where we're trying to describe and then tackle some of these really big and really interesting questions about how life is structured and how life has evolved, how the patterns that we see have evolved, why Brazil is hyper diverse and why Canada is low diversity. But it's not the only example of this. I think this is perhaps a trend, and I think it's an important trend to consider. Tiago Rangel has a very nice paper in Science, a very different model, but again, simulating species evolving over time and the distribution of species evolving over time to see whether observed patterns can be replicated for bird richness, for example, in South America. So it's a much finer resolution model. It's a regional model. They really focus on niches and species distributions, and then look at how the climate has evolved over time and the evolution of patterns through time. It's a very nice paper, and I think it's really great that you have different models using different approaches, which can then help to inform one another and improve this at multiple scales. So really what I suggest is that maybe we should start thinking more about a mechanistic biodiversity science, where you start with your data gathering, your biological data. And again, we're getting more and more data. The methods for getting data are more sophisticated. And then you develop, or you fit statistical models to these patterns. So you fit curves, you fit regressions, and that allows you to generate hypotheses. So maybe you find a relationship with temperature. What are the mechanisms? Are there multiple mechanisms? If there are multiple mechanisms, you could start to develop mechanistic models of these processes that actually simulate those processes in detail and use those to test these hypotheses that you generate using statistical models. And then you can feed these back to help inform your biological data gathering. Maybe you need more information on mutation rates or similar. But I think it's important to think about, given that human impacts dominate what's going on in the present day, why is this important? Well, for one thing, it's important because this is one of the most fundamental questions of life on Earth, and one of the most interesting questions, right? We have this incredible biodiversity. It's a key feature of our planet. How was it formed? How will it evolve? I think those are crucial questions. But I know for myself, most of my research now focuses on very applied questions, because we are entering this era where human impacts dominate everything else. So this is a recent study that we looked at benthic foraminifera, these planktonic organisms, and we looked at how the shape of the latitudinal gradient had evolved over time. So if you look at the last glacial maximum, you really have a tropical peak in the relatively recent past. But then as we move to the pre-industrial, we start to see a strengthening of the subtropical peaks. And if we take this information and project how it will look in 2090, we see a real decline in tropical biodiversity, and again, a strengthening of this bimodal pattern. And in fact, if you look at 2090, the evidence suggests that the diversity in tropical systems may be lower than it's been for millions of years. So really trying to understand the factors that affect biodiversity, and especially whether they operate on evolutionary or ecological timescales, tells you something about how quickly climate change will impact systems, and more importantly, what the recovery times will be. Excuse me. So as a very brief conclusion, I have described a process-based model of global biodiversity patterns that has very few parameters and simulates community, doesn't fit statistical curves or lines. We find that temperature and area or resource limitation give a good fit to empirical data. And broadly speaking, evolutionary processes, from our model anyway, seem to be more important than ecological processes in setting these planetary scale richness patterns, but ecological processes may be important in terms of the identities of the species that are found in any particular place. And then perhaps, given this and other work that's going on, we're moving towards a more mechanistic biodiversity science, where we explore some of these mechanisms through models and other approaches. And with that, I just want to dedicate this talk to my former colleague, Ron O'Dore, who was on the Biota program external committee from 2011 to 2014. And I believe he came to Brazil and participated in several meetings with you. Ron was also a colleague in the Department of Biology at Dalhousie, and was, as you know, the chief scientist of the Census of Marine Life. Unfortunately, he passed away this year from COVID-related complications, but he's left a profound legacy. I mean, for myself, he was very important. I really got my start with the Census of Marine Life, which was a big and long-term program like Biota. And I think Ron opened many doors for many people. So I think we should remember that and remember Ron and his contributions. So with that, I thank you and turn it back over to Professor Joly.
Well, Derek, thank you very much for a very inspiring talk. I think it's quite difficult when we have to use all this information that you use it, not only in terms of the diversity, but also in terms of the evolution of this diversity when it comes to set rules for conservation areas, marine conservation areas. Usually you take the consideration the on the top of the sea, the plankton one, and perhaps still pelagic, it's what considers, but you never consider the deep diversity. So we might getting a lower representativity of deep sea biodiversity in those conservation areas. And thank you also for the tribute you made to Ron. It really was a great help to us when we were developing this marine area within the Biota program, and was a very good fellow. Having him here for the meetings was very nice. So now we start the session where we have the questions. First of all, I'll give you the opportunity to Mariana and Alex if they want to make a questions to Derek. And I have to remind people that are on the YouTube that you have to send questions to contato@biota.org.br. Now, Mariana, the floor is with you.
Okay, thank you very much, Jolie. Thank you very much, Derek, for a very inspiring presentation. I do have a question. I'm not sure which is the current knowledge of what is the unknown biodiversity in the oceans. I think the last numbers that I remembered is that we know about 10% of the biodiversity in the ocean. I don't know if that's correct, but I was wondering how this lack of knowledge of marine biodiversity impacts our ability to modeling this diversity in the oceans.
Yeah, I think so. It's been a while since I've looked at the numbers, but the last time I saw about 300,000 marine species were on the WORMS website, which is the World Register of Marine Species, where that information is compiled. And there may be, well, we estimated maybe 1.2 million marine species. So actually the proportion of species described is I think, reasonable and growing very fast. But of course, it's very different for different environments. So the deep sea is incredibly undersampled, and it's very easy, I think, to find a new species in the deep sea. But the limitation is the rate of describing these species in the literature. And so I think when you think about how it affects modeling, I think that's less of an issue than how it affects conservation, because I think if we're making conservation decisions and decisions about the where to place marine protected areas, for example, we need to be careful that we try to include unique systems that may have high biodiversity that hasn't been properly described yet. And I think the deep sea is an example of that. And I think that, not just from a protection perspective, but from a ecosystem service perspective. I mean, I think the deep sea is a very unique environment that has organisms that have adapted to very extreme conditions. And so for things like medicine and drug discovery, there are estimates of huge value of potential, for example, anti-cancer drugs from deep sea organisms. So yeah, we just need to be mindful of that, I think. But I'm not sure that it's any worse than terrestrial environments where many millions of species may also be undescribed. So yeah.
Thank you.
Mariana, we have a question for you and Alex. A question that was made by Mary Gazala. She said was congratulations on the presentations, but she was worrying or wanted to know if any legislation was produced based on the Biota results for coastal and marine areas. Perhaps Alex would reply to that.
Yes, sure. Thanks for the question. Yes, it's an ongoing process, since there is a timing of the discussion and implementation of public policies. But we can list some examples. One of the most recent example is related to the management plan of the marine protected area of the northern coast of São Paulo. And some of the outcomes of the Biota Araçá project were incorporated into the mapping, into the zoning of the protected area. For example, Araçá Bay, it was not defined in the decree that created the protected area, and then it was mandated to the management plan to define the area. And then it was the PhD of a student of mine that discussed it with society, with researchers, with local people, fishermen, other users, and then we produced an interpretation based on different views of what would be Araçá Bay, which is not limited by the opening of the bay, which is interesting. So people understand that the bay interacts with the channel based on several proxies, several information, and then the map that was produced by this collective exercise, where a lot of social learning happened, was incorporated into the management plan of the marine protected area. So this is one example. Of course, there are several other examples related to fisheries, not necessarily made by the Biota Araçá project, but by others, or even, for example, things that are related to biodiversity, but are more focused on environmental impact assessment. For example, the fight against the combat of marine litter, or other threats that face biodiversity. So there are several initiatives that are strengthening the usage of the resources, of the results, of the outcomes of the projects in public policies, and not only public policies, but also in the private sector. Thank you.
Okay. There is another question here that is related to your talk, Derek. People are asking, does this theory and the book and the papers that you have been producing being used for this large international organisms like FAPESP or the Convention on Biodiversity to establish or to change recommendations on sea and ocean?
So the modeling work, no. I think the modeling work is more understanding how patterns of biodiversity came to be. I think it's important in a conservation context, as I mentioned, because if recovery is on evolutionary timescales, it's very different from recovery being on the ecological timescale. So I think understanding how those factors shape diversity is important. I think, to be honest, I think the things that really shape or have shaped conservation and understanding and biodiversity are the maps of spatial distributions. And as I say, those go back many years, but they've recently evolved and they continue to evolve. And we're starting to see now more high resolution model estimates of diversity. So those are the things that really help to inform conservation and scientific processes.
Okay. There's another question here for you by Dr. Wellington Araújo. How to separate the temperature effect on metabolism of the study species from the effect on the organisms associated to primary productivity, which in turn results in a secondary effect on the study species?
Yeah, so the temperature effect on metabolism operates just through interaction rates and speciation rates. Productivity operates through setting the availability of resources. So you can have a low productivity, high temperature area where metabolic interactions and rates are faster, but there's a real constraint on the number of organisms that can be supported. And in the model, you can switch on and switch off these effects. So you can turn off the temperature effect and turn off the productivity effect, excuse me, and see which one or whether both in combination better reproduce patterns. The other thing I would say is that the marine environment is really interesting because terrestrially, I know there's more overlap between temperature and productivity. Not completely, but they're more highly correlated. Whereas in the oceans, the areas of high productivity are much more separated from areas of high temperature. So it's an interesting contrast between terrestrial and marine environments in that case.
Well, thank you very much. I have to ask Mariana and Alexandre if they have any final comments to make.
Yes, I would like to reinforce, based on Derek's presentation, which was brilliant, about the importance of modeling. Some more comments, but it's really important to, in my opinion, to be fostered in the Biota program, to build capacity to work with models and to understand the limitations and the potentialities of models, and mostly how modeling could or should be integrated with other disciplines like genetics, like evolution, like omics, in a way to integrate, for example, what you were saying about changes in patterns due to climate changes and the evolution, the evolution of species that will change their genes in proportion to that will allow them to live in a warmer water, for example. So this is really, really important to couple. And then I think we have a very good room. We have room to expand this approach. So thank you very much for bringing this and reinforcing the importance of modeling and the coupling with other disciplines.
I just want to point out that I also agree with Derek that the lack of knowledge that we have on marine biodiversity has to be, in a way, overcome by other tools because we need to focus on conservation. So we do need to very fast move into modeling and using other tools for the proxy of biodiversity, and to be able to pinpoint the locations that need to have conservation, because changes are happening very fast, and we have to move very fast also to protect this biodiversity, even if it's unknown. So, thank you very much.
Well, I'll thank you both, Mariana and Alexandre, for taking the responsibility of summarizing the progress or the impact made by the Biota program in the coastal and marine research. But especially would like to thank Derek for accepting our invitation and presenting a very inspiring talk. Last year, I've organized a school for modeling. We had Caroline Lundquist here as one of the persons involved. She brought some of the coastal marine modeling that she worked with, but your talk today brought this up very strongly and how important it is. So thank you very much for taking part in our anniversary webinar.
Thank you very much. I appreciate the invite, and I want to congratulate you again on your program, and I shall enjoy following your successes in the coming years.
Okay. Well, thank you all. We will finish here our transmission. And again, I apologize for the delay we had in the start. Estamos aqui terminando a nossa transmissão para o YouTube e eu gostaria de novamente me desculpar pelo atraso que, por razões técnicas, nós tivemos no início dessa transmissão. Muito obrigado a todos. Espero reencontrá-los no dia 8 de outubro, quando nós teremos o 5º webinar, que vai ser focado em taxonomia e ecologia animal, os avanços no conhecimento e impacto que o programa Biota trouxe para essas áreas. Bom dia para todos.